Okay, we're here, we're live. It's Monday. Oh, Monday. We're here on research in Manoa, thanks to Talia Ogliori of the Chancellor's Office, who helps us with guests. And our guests today are Greg Stewart, who's professor of oceanography in SOAS, and his office and laboratory is in Seymour, the Center for Microbial Oceanography Research and Education. See, I know that. Welcome to the show, Greg.
Thank you.
And we have Emma Nuss, and she is a graduate student, and she works with Greg, am I right? Okay, so you guys know what each other is doing. Okay. And we're going to call this show Antigens, I guess that's right, Antigens in the LOI.
Or about pathogens.
Pathogens, pardon me, pathogens. What's the difference?
An antigen is just a protein epitope that an antibody, a part of the immune system, will recognize on a foreign invader or anything you eat. So it's a recognition for an antibody, recognition protein.
Okay, so a
we're talking
pathogen
about,
will have antigens.
That's right.
But an antigen will not necessarily have pathogens.
That's right.
Okay. Okay, and you guys are doing research on these pathogens in the LOI, namely Vibrio vulnificus.
Yes.
Which we're going to talk about in some detail because people have to know about that. But first, let's see what your identities are. So, Greg, you went to Scripps, or rather, you took your PhD at Scripps in San Diego.
That's correct.
What was that like? Did you enjoy that?
Oh, it was a dream come true. Yeah. It was great.
What made you do that?
I started out actually in a very different field. I was in I studied neurobiology as an undergraduate, and after graduating, got a job as a technician in a medical-related research lab. And my other love had always been the ocean, but I went down this other track, and after doing that for a while, I realized that what I wanted to do day to day was get outside more, do more field work, do something more environmental. And so that's when I decided to switch tracks and get enrolled in the marine biology program at Scripps Institution of Oceanography.
That's a really first-class place, isn't it? That's top of the line.
Yeah, it was a wonderful experience, and it's a dream to go there. And once I got there, I grew up on the East Coast, so California was just sort of a vision for me.
I had the same experience, as it were, years ago.
Yeah.
I saw California as a kind of a cartoon.
Yeah. Yeah.
So, what you said makes me want to ask, is when you're into science and you're trying to find your niche, you have to keep on thinking of the options. You can't just settle down to something right away. You have to find something that works for you. You have the flexibility to do that, right?
And that's something I try to stress to the college students that I come in contact with through teaching and mentoring, is to not worry too much. What you study in college isn't going to define you necessarily, that you have options and flexibility. And sometimes it's very helpful to study one thing and then later switch, because you can bring a new perspective to a different field.
Yeah, become multidisciplinary. But you have to be in science to get into that world, right?
That's right. And so that's the key thing I tell them, is don't worry so much about specializing too early. The key is to get a really solid foundation in all the basics, the fundamentals of biology and chemistry and physics.
Yeah, God, I wish I had more of that. In high school biology, I loved it, and then I went to law school. Anyway, the other thing I wanted to ask is, by what age? This is a hard one. By what age? I know it's relative. It's all different for different people. But by what age should the scientist feel that he is or she is settled down? In other words, it's like getting married, I suppose. That age changes with our culture. But it could be, older or younger. But by what age would you expect, as a career pattern, that somebody will have fixed the direction of his career?
I think maybe at retirement.
Touche.
I think it happens a lot that we do get into a track, and there are just so many different paths. I mean, there are some people that have been studying the same very narrow thing for a long time, And that's a good way to go. They are recognized as the expert in that thing. And so I think, focusing is just one of a number of paths, and that makes you the recognized expert. But there are many people too who have been more Renaissance type people who jump around a lot and are very successful at it. And again, I think that cross-fertilization of what they've learned in one field, taking it to another field, can produce some really creative and productive science. So I don't think there's even a recipe, once you're out of school, for what the best way to proceed is.
And a lot depends on the people you're with, isn't it true? I mean,
Oh,
you
sure.
don't live in a silo. You're talking to people and seeing their work, not only immediately around you, but continents away.
Yeah.
That's because that's your community. I just wonder how you feel Seymour fits in all of that for you.
Well, Seymour's been great. So yeah, you're right. The colleagues that you have around you makes a big difference in how productive you can be, and just the sort of the intellectual stimulation that you get is very important. And that's one thing I liked about Scripps, and one thing I like about being here in Hawaii as well, is they're large departments. There are a lot of people concerned about the same topic, and it creates this critical mass that really feeds the creativity. And Seymour is also a large entity. Not all of it's here. It's headquartered here, of course, but we have partner institutions and a number of other institutions on the mainland. So we have a broader community. So it's a lot of people, this critical mass of people all concerned about the same kinds of things, and it really helps with creativity and thinking of new research directions, and allows you to do things you wouldn't be able to do otherwise.
Yeah. And Hawaii and UH Manoa has to be important in the world of marine biology, because we have the marine and we have the biology.
That's right. Yeah, this is an excellent place for oceanography, marine biology. As you pointed out, you can't ask for a better location. Right in the center of one of the largest ocean on the planet, so it's a great place to be, and that's why we get so many talented people coming here to study.
I've noticed that, exactly that, yeah. Let's turn to you, Emma. You're a graduate student on your way to what? You're going straight for a PhD?
Working on my master's first,
Okay,
but
master's
potentially PhD afterwards.
Okay, and where did you go to undergraduate school?
UC San Diego, actually.
Okay, so you know about Scripps.
Yeah.
Okay, and why exactly did you want to do this?
Well, originally being in San Diego and having Scripps as an opportunity right there, I started taking oceanography classes, and so that got me interested in going to grad school in oceanography. And then once just applying to schools and finding out about different research paths, then I met, through applying, I met Brian Powell, the professor at oceanography that I'm working with as my advisor for my thesis project, and he brought up this project about working on predicting pathogenic bacteria in the alloy. And so through that, I found out about the research and the ability to be able to predict concentrations and hopefully limit risks of infections of this bacteria. What drew me to it.
Sounds a lot like medicine. Did you have aspirations of becoming a doctor?
No, not at all.
You just want to be a research scientist, eh?
Yeah, maybe.
Okay, all right. So, what is your master's in? I mean, what's the thesis?
My thesis is in physical oceanography, and particularly in building a coupled physical biological model to predict concentrations of Vibrio vulnificus in the alloy.
There we go. So you're working, and we're here to talk about your master's thesis. Okay. And it's just in case anybody didn't catch that, it's Vibrio vulnificus. It sounds awful, doesn't it? How awful is it?
Most of the time, it's actually rather charming.
You remind me of an aquaculture expert in Hilo, or rather in Kona. And I said to her, she grows what do you call an aquaculture product, I said to her, you know, do you ever eat this product? She says, no, they're my friends. They're my friends, so I could never eat them.
Yeah.
So are you saying, Greg, that these bacteria are somehow your friends?
That might be a little too far. I will say that I have developed a certain fondness
Okay.
for them.
I
That
haven't
may
seen
change
this
if I
before.
ever contract an infection from my studies.
They would never hurt you. They know who you
Yeah,
are.
we have an understanding. Yeah.
So who are they exactly?
So these are bacteria that belong to a rather broad group, a genus called Vibrio. So there are lots of Vibrios in the marine environment. You find them from fresh to full-strength seawater. But a lot of them are, you tend to find lots of them in estuarine environments, brackish water, where fresh water enters the sea. So places like the Canal, where you've got a mixing of fresh and seawater, those are the locations where you tend to find a lot of these
They can
kinds of
exist
bacteria.
in the fresh or in the
Yeah, some of them,
the
they
brine.
all, a lot of them are halophilic, so they like at least a little bit of salt, but some of them can tolerate lower salt concentrations than others. And that's one of the techniques you can use to try to distinguish what kind of Vibrio you have, is their salt tolerances, although that doesn't get you very far.
We're going to take a look at a picture one.
Yeah.
Let's see. There it is. There's a picture.
Yeah,
of a,
see? Isn't that cute?
So can you tell us what we're looking at here?
Yeah. So this is a picture of Vibrio vulnificus. It's a bacterium, gram-negative bacterium in the gamma proteobacteria. Not that that means anything, but it's very small, so we're looking at a few microns long there. It's sort of sausage-shaped, and you'll see at the very end, you might be able to make out a very thin thread-like structure,
Yes,
which
we can
is
see that.
the flagellum.
What does that
and it
do?
uses that to swim.
Ah,
It's actually, yeah, they're amazing structures. The flagellum is this thread-like protein made out of flagellin, and it's attached into the membrane of the bacterium, in this case at the very end of it, attached to a little protein rotary motor that just spins around based on a proton gradient. Just drives that rotary motor, which flips that flagellin thread around in a circle, and you see how it can be kind of curved, and it just propels it through the water.
you know, Dave Carl and I were talking about those examples of kinetic movement
Yes.
in microbiology,
Yes.
which someday conceivably could be used as a source of energy
Yeah.
with nanotechnology.
Yeah. Yeah, it's
really
it's
exciting
fascinating to think about.
that.
Here are these absolutely tiny little rotary motors just spinning around super fast, all made out of protein.
You have to figure out how to do that.
Yeah.
But that's not your scientific
Yeah.
research right now. Okay, so we have the vulnificus. Is it likable? I mean, is there anything redeeming about it? I mean, aside
Well-
from whatever your relationship is.
Yeah, I think my personal feelings aside, yeah, it's a natural component of these types of environments. So that's one difference between this particular pathogen that we're studying versus a lot of other studies of pathogens in coastal waters. What you're actually looking at are pollutants. They're human pathogens that get into the water because they're in the sewage, coming from humans, getting into the sewage, and then when you have poor treatment of the water or leaky sewer lines or overflows, then you have the sewage leaking into the system and it carries with it potential pathogens. But that's a contaminant. What we're looking at with the Vibrio is a completely natural part of the ecosystem. So they're out there. Their numbers aren't super high compared to the total number of bacteria in the alloy, which is enormous. I mean, we have in a milliliter, so like in a teaspoon, you may have 25 million bacteria. And these, maybe you'll have 50,000 of them. At the high end might be these pathogens.
So they're relatively less populous.
Yes, but that's in part because there is so much bacterial diversity out there.
Yeah, we don't realize we live in a world of
Yeah.
millions and billions all around us, including our bodies,
Yep.
including, for example, this I know it's hard to say, this unnamed beverage
Yes.
product that I have here. And speaking of
Caramel-colored
which,
beverage.
I'm going to take a sip of this during a break. We're going to
Yeah.
have a break now, so I can check out the levels, okay?
Yeah.
That's Greg Stewart. He's a professor of oceanography in SOEST at Seymour. And Emma Nuss, she's a graduate student also in SOEST. Yeah. And we're talking research in motion. We're talking about pathogens in the alloy. We have a long way to go because there's stuff here that will really stick in your brain. We'll be right back.
Aloha, I'm Kili'i Akina, president of the Grassroot Institute and host on Ehana Kako, a weekly program on the ThinkTech Hawaii broadcast network. Ehana Kako means let's work together. Think of the sad alternative, let's not work together. Here in Hawaii, with all of our diversity and the richness of the people, it's important for us to come together around issues on the basis of what's right and what's good and what's going to serve the common good. And that's what we try to do at Ehana Kako. Every week we interview movers and shakers, people in government, business, and other sectors of society to talk about how to create together a better government, economy, a better world here in Hawaii that can bless the rest of the world. I thank you for your attention to ThinkTech Hawaii, and we look forward to seeing you every Monday, 2 to 3 p.m. on the ThinkTech Hawaii Broadcast Network. We're Ehana Kako, and we wish you well. Aloha.
Okay, we're back, we're live, we're here on Research in Manoa, talking about pathogens in the alloy with Greg Stewart, a professor of oceanography at SOEST, and Emma Nuss, a graduate student, and they're both working on this vulnificus, Vibrio vulnificus. I may never forget. I may never see a glass of water where it doesn't remind me of this conversation. Okay, so in the alloy, we have, it's a natural phenomenon. We have the vulnificus.
Yep.
And it was there at the beginning of the alloy. It's where the brine meets the fresh.
Yeah, pretty much anywhere in the world. The only exceptions are these tend to prefer warmer waters. So the further north you go on the coastlines, you tend to see fewer of them, or the only times you see any significant amount are during the summer months.
Yeah, but warm
They
is
like
an
warm
operative
water.
term.
Warm,
Okay.
yeah.
And that takes us to the summer of 2006,
Yeah.
which we need to talk about here on this show about vulnificus. Vulnificus was the star villain,
Yeah.
the star villain of the summer of 2000. What happened?
Yeah, so there was an individual who, I don't get into the details of how it happened, but he ended up in the alloy, had cuts on his legs that were not taken care of, promptly cleaned, and ended up contracting. They thought in the beginning that it was the so-called flesh-eating bacterium or a streptococcal infection, but it turned out upon testing that he was infected with Vibrio vulnificus. And because of the long delay and other extenuating circumstances, there were a number of risk factors. This individual would have been particularly susceptible because of cirrhosis of the liver. So that, coupled with the delay in getting treatment, failing to wash out the wounds immediately after exposure, ended up contracting the infection that turned into a septic infection, which means the bacteria then got into the blood and were growing very rapidly in the tissues and the blood, spread throughout the body. And it was truly horrific.
They couldn't save him.
No, and they tried desperately amputating limbs to try to stem the infection. But
Keep ahead of it, but
yeah,
they couldn't.
No.
You know, people think that antibiotics are the panacea, that they can save you from any infection.
Yeah.
Not really true.
Yeah, it depends on whether the pathogen itself is susceptible to the antibiotic that's being used, and the second thing is getting the treatment in time. If things have progressed too far, then you have the body also begins to fight itself with inflammation and
Yeah, everything goes haywire.
yeah.
Well, before the show began, we went and looked on a browser. We looked up, vulnificus, and and images, and my goodness gracious, there are some. This is a family show. We could never show you these pictures.
They're
It's gruesome. It's as gruesome as anything you've ever seen. Your body is coming apart right in front of you.
Yeah.
Awful.
Yeah, a lot of these infections. So there are different ways you can become infected. On the mainland, one of the main routes of is through ingestion. These things grow in brackish water, which also happens to be where a lot of shellfish are harvested and grown.
You can get them from shellfish.
Yeah, the shellfish filter things out of the water, and they tend to accumulate a number of pathogens, including things like vulnificus, so they can reach higher levels in the tissues of the oysters. And if you consume uncooked oysters, you can be ingesting an inoculum of pathogen. And if you are particularly susceptible, then you could end up getting an infection that way. And mostly those will just be gastrointestinal distress, and maybe some vomiting and diarrhea, which will clear. But people who are particularly vulnerable, it can end up, again, getting into the bloodstream. And once it gets into the bloodstream,
cut somewhere, some kind of area where you've been cut in your intestine, I'm
like
sure that would get into the bloodstream. Yeah.
And another route for infection, which is more common here in Hawaii, is through recreational water exposure. If you're in brackish water and you have an open wound exposed to the water, then there's an opportunity for the bacterium to get directly into the tissue.
And that's what happened to that fellow.
That's what happened in that case. Yeah.
Well, let's just take a moment and let me ask a question, to Emma. So this is the thing you want to study? You go through school, you study, you work hard, midnight oil, take tests, whatnot, write papers, and this vulnificus is how you want to dedicate your research? Why?
I think, as Greg was mentioning, the fact that a man has died and that other people, if they got infected and were susceptible, could also sustain infections, and that being able to have a better understanding of the distribution of the concentrations of the bacteria would allow to limit the risk of infections, and so being able to understand that.
Yeah, well, let's examine that. Well, why do you say that if you can figure out what causes high concentration of Vibrio vulnificus, then you can somehow save people from it? What's the connection there?
So the connection is, we know that temperature and salinity determine growth of this bacteria, and so being able to understand the distribution of temperature and salinity in the aloi and the surrounding South Shore will give us a better understanding of where this bacteria would be able to grow.
Okay, so you want to find out where it would grow, and then you want to take those remove those factors so it doesn't grow.
Not so much to remove the factors, because it's a naturally occurring bacteria, but to be able to understand where the risk of exposure,
Okay,
and
so
then
you can warn people
and
treat them more quickly. Okay, so you don't have to touch this, do you?
No.
In fact, you don't touch any of these biological materials.
I don't.
So how can you do research in vulnificus without touching them?
So my research is in using computer models to simulate the physics involved in controlling the movement of water in the aloi and along the South Shore.
Okay, but all this computer model, I'm learning, by the way, from SOWEST. To make a computer model, you need data.
Yes, lots of good data.
Where are you getting your data from about vulnificus?
Well, Greg is
Oh,
the main
now
person.
there's a connection. Okay.
For the biological side, we are doing so we've isolated hundreds to thousands of strains of this particular bacterium from the aloi, and we try to measure their growth rates in situ in the aloi. That's what we're trying to do now, but we've also done a lot of experiments in the lab where we isolate them, grow them, see how they respond specifically to changes in temperature, changes in salinity, how that directly affects their growth rate. And then we give that information to Emma, and she puts that into a biological model, which says, okay, if we've got this temperature, this salinity, they should be growing this fast. But then needs all the other data. Well, what's the temperature and what's the salinity in the aloi? So we have other sources of data to get that, and then we try to merge them all. And we would talk about the data from the physical chemical data.
Yeah, let's talk
Did you
about the
use-
physical chemical data.
Well, we're using a regional ocean modeling system, and so the data from there, I think it's a nested model, so we're looking at a fairly small region, just including the Alawai and a little bit of the South Shore, but it's nested within larger models that we take the original, I think there's a global ocean model that the data from there gets scaled down into our small model of the South Shore.
So is this for verification of what you found from Greek's data, or is this?
This is more for getting data for knowing how the ocean is, because we're looking at a time period of 2008 and 2009, because that's when we have data of the Vulnificus and the Alawai. And so we're looking at like previous physical data from then, so we can hindcast concentrations of the bacteria. And so
She's going to deliver your conclusions, right? You're
I'm-
giving
I'm
her the
waiting.
rubber together, yeah. So do you have any idea what how long have you been working on this?
been here for a little over a year and a half.
Okay, so then you should have some idea where it's going. So where is it going? What do you think your conclusions are going to be?
Well, hopefully the conclusion will be that our physical model is able to fairly accurately predict what we already knew of the concentrations in
So
the
what you
Alawai.
thought on a deductive basis is true on an inductive basis.
Exactly.
And what is it? I mean, what are the big factors that you think are going to pop out of this? Heat, salinity, I don't know what else. Anything else?
Temperature and salinity are the main ones, but also the advection of water, because when there is a big rain event on the South Shore, a lot of that water in the Alawai gets flushed out. And so looking at, in our physical model, looking at how that affects the concentrations of the bacteria is also
And those
crucial
are
part.
all factors that were present when Mr. Alawai fell in the water.
Heavy rains.
Heavy
Yeah, so
rains.
rainfall has a,
And I misspoke when I said the summer of 2006. It was the spring, wasn't it?
the spring.
The
yeah, I
40
think it's
days of
April
rain we
and-
had,
Yeah,
yeah.
exactly, yeah. March into April, and I think it was early April
Yeah,
that the
it
incident
was
happened.
really too long. We can expect
Yeah.
more of that with climate change later, but that's another discussion. Maybe
Which
it's
is a relevant.
early warning.
Yeah, I mean, that is a relevant one, talking about predicting the risks from Vibrios in particular, is a lot of folks have been questioning that. How are rising temperatures going to influence the risks from things like Vibrios, because they love the warm temperatures. And so we may see areas where Vibrios are a problem creeping northward along, on the mainland. And it may also be influencing concentrations in the future here in the
Yeah.
islands.
So don't go in the water, actually. So how did it work then in 2006? You had 40 days of rain. We were all getting really tired of that. How did that affect the temperature, and how did that affect the salinity? I mean, something happened to the Alawai, right? These natural forces were a little unnatural that spring,
Yeah.
and they were creating an unnatural, setup in the Alawai and coming out of the Alawai. This was at the mouth of the Alawai.
That's right.
So what happened exactly to the water, and how did it happen?
Well, one of the unfortunate things is that we had been studying Vibrio vulnificus at that time, but at that time we actually had a project studying it on the mainland following Hurricane Katrina. So we were in Louisiana, Lake Pontchartrain, doing a study there, because following Hurricane Katrina and the disturbance from that, there was a big surge in Vibrio infections and deaths post-Katrina. And so we had funding to go there.
In that area?
In that area,
Louisiana.
specifically in that area. So
Yeah.
we had funding to go there. And it was in the middle of that study, we were in the midst of that when this incident happened here. And that was kind of
It's kind of a scientist's dream, isn't it? You're studying something, a set of phenomena over here,
Yeah.
and then now you have the chance to compare it
Yeah.
against the similar set over there, and you can learn by the comparison.
Yeah, and it made us realize that this was a good, interesting project that we were doing on Louisiana, but here we have this issue right in our own backyard. So at that
Hurry
point,
back.
Yeah, we hurried back and initiated a quick study, and from there have tried to keep funding going to try to sort out the ecology of this bacterium right in the waters that are closer to home.
It's really so interesting. This is a scientific research project that is actually driven by an actual event.
Yeah.
Or I mean, that captures the public imagination like few other things do. Let's take a short break. We'll come back, and one of the things I like to pursue the issue of exactly what the changes were that made the concentration of the Vibrio so great enough to be, in that case, fatal, and also to compare what happened there with other cases we've seen involving man-eating or people-eating bacteria, which we should all know about that. Okay, let's Greg Stewart, professor of oceanography in SOWEST, with his laboratory in Seymour, the Center for Microbial Oceanography and Research and Education, and Emma Nuss, who works with him. She's a graduate student. She's a computer person, and she's modeling. She's like a model, She's modeling. She's modeling all about these pathogens in the LOI. We're calling it here in research in Manoa, and specifically we're calling it. We're looking at Vibrio vulnificus, which is a term we should all be familiar with now. We'll be right back.
Hi, I'm your host on ThinkTech Asia, Bill Sharp. I look forward to you joining us each Monday between 4 and 5 o'clock when we film right here in our studio in downtown Honolulu. The show, ThinkTech Asia, focuses on contemporary events in Asia, and by Asia we mean anything from Hawaii, south to Australia and New Zealand, west to Pakistan, and as far north as the Russian Far East. Clearly, this is one of the most economically dynamic centers of the world, and we bring you up to date on what's going on in a whole host of countries in this very vital region. We look forward to seeing you. Aloha.
Okay, we're back. We're live. We're here on research on Manoa pathogens in the aloi, namely Vibrio vulnificus. I'm getting it. With Greg Stewart, professor at SOWEST, and Emma Nuss, a graduate student there. So going back to the confluence of events and forces and factors that happened around Mr. Aloi's death, what the water obviously had a higher concentration of vulnificus, and that was fatal to him.
We don't know
Okay,
that the concentration was higher.
you don't know. Okay.
We don't know that.
So
And that's part of the disappointments, is that we hadn't been studying it up until that point. It was this event that set us onto this study of going out and making the measurements, and we would love to have had samples prior. But then we set about doing a year-long, more intensive study of the concentrations to see if we could infer, based on what we learned, were they likely higher or not. And we think they probably were higher.
because of the year in which you did your subsequent examination of the concentration.
Yeah, and based on...
But you could not have achieved the same 40 days and 40 nights.
No, we may not see that again for a long time.
Good.
Yeah. But just looking at what we did learn was how the Vibrio respond to rainfall events. And what we found is that heavy rainfall can have competing effects. We said how the Vibrio tend to like water that's not so salty. So when you have high rainfall, you got the runoff into the canal, it drops the salinity down into a range that the Vibrio particularly like. So that can stimulate their growth to higher levels. But at the same time, that rainfall flushes out the canal. And so there's this mid-range where, some rainfall tends to promote the growth, but when you have really heavy rainfall, it just flushes everything right out. And you can have essentially fresh water in a lens on the very surface, and there are hardly any of Vibrio vulnificus growing in that. So it's got these competing effects.
So do we know exactly where the the optimal point is in terms of salinity in fresh water?
It's around 10-ish.
That's where they really like to be.
Yeah, so freshwater we call 0. Full strength seawater out there is 32, 34. And then something around 10 to 20 is right in that mid-range is where they really grow the best. But you can still find them in saltier water as they get washed out. And that's part of the trick that we're relying on Emma's work to help us deal with the fact that the environment is constantly changing. And things like the rainfall changes the salinity, but it also changes the rate at which they flush out. And so that's why we're hoping this, by teaming up and collaborating, putting the physics together with the biology, we can do a better job of predicting than we've done in the past.
Yeah. So if all be known, if your model, is very precise, what would we do with the model? What, would I, for example, would I have a vulnificus alert and say, you guys better be careful?
Well, we sort of have that
You already have
vision,
that.
which we have the vision of what we would like it to do. The question is whether we will ever have the accuracy and precision to pull it off. But we already have implemented so we're also using data from the Pacific Integrated Ocean Observing System, which is part of one component of these larger global ocean observing systems that get set up. So we have one headquartered here in Hawaii on Oahu at UH being administered there. And it's a suite of sensors in the near shore and some right at the mouth of the Canal that feed us continuous data,
It's just live.
temperature, salinity, yeah, it's live on the web, turbidity. And there are other sites, and it's a website you can go to, PAC-IUS for Pacific Integrated Ocean Observing System. It's feeding data from all these
That's the thing I don't
platforms.
think people fully understand. You're not out there with, test tubes and cages and,
Well, we have to do that
oh,
too.
you do that too?
Because there are many things, the physics and chemistry we can do with sensors, which is great. We can make an ocean observing system, put sensors out, spread them around the coastline, and it's just the data streams
And
in.
you make them public. Anybody can look at them.
We don't have a pathogen sensor that can sit there and feed us real-time data.
Could you build one?
People are working hard on building them, and there are prototypes. There are certain types of systems that you can put out there that basically replicate all the hard work we do in the lab and make it roboticize it and put it into a can that floats in the water. But they're really expensive, which means you can't buy very many, which means you can monitor one spot. So we need the cost to come down. Probably we need methods improvements. The methods we have right now are too complicated. We need faster sensors. There are prototypes, ways of doing it, but the big problem we have is the signal-to-noise ratio. That's the biggest hurdle for putting a sensor out in the water that will tell you in real time how many pathogens are there. As I mentioned before, the Alawai's got 25 million bacteria in every teaspoon, and a tiny, tiny percentage of those are the things that might actually hurt you.
Yeah.
So the pathogens are a tiny
I
percentage.
suppose I'll make a wild, unscientific guess, but so you get a camera,
Yeah.
it's an underwater camera or a camera, underwater camera. It sees through the water, and it's going to be a very microscopic camera. It's going to see, within a very small sample, and it's going to actually take a snapshot of. Okay, then you have bacterial recognition, like face recognition and bacterial recognition, and it actually counts the number of these vulnificum, vulnificusum, whatever.
Vulnificus.
Vulnificus
Yes.
people. I think I know some of them.
Yeah.
And then it sends you a report back by radio.
Yep.
This is our sensor that counted what the snapshot had.
Yes.
Shows X units, and we're going to go back in another 20 minutes and do it again.
Yeah.
Would it be something like that?
An analogy to that, probably not a microscopic camera looking at the cells. As you saw that when we showed that cell, the electron micrograph, it's sort of this sausage-shaped thing. And there are a lot of bacteria out there that have that shape. So
On top of each other.
So it's really hard to look at a bacterium and know for sure what you're looking at.
Yeah.
And I would say even for the vulnificus, another major challenge for us is that not even all the vulnificus are the same. There are certain subtypes of vulnificus, some of which are more prone to end up causing disease in humans and others which are less so. So not only we have to look for vulnificus, we want to know which kind of vulnificus.
Can
And
you
it
tell
gets
the
tricky. And
difference
really
with-
we need molecular biology to do it. So we can't, which makes it a little more
That
tricky.
means you have to have a laboratory. You have to test them in some way.
Yeah, or we need to find the right signature, maybe but it's still going to be a molecular recognition, nothing we can do visually.
Well, that all takes me to, to this. You guys are now dealing with one microbe in a world of trillions, gazillions, and googles of microbes of every
Yeah.
kind and every head of every pin. But you must be learning things here with vulnificus that you could use with other bacteria, other microbes, other pathogens. Can you give me a direction on how that evolves for you as a scientist?
Well, I think one of the things we're trying to pioneer with this study is to try to develop a predictive system. So I mentioned the PAC-IUS and all the sensors. The reason that's important is because they sense things that we already know how to sense. We can get sensors off the shelf. We buy them, we can put them out there. That technology is very well developed. We just have to put it out there. But it only tells you temperature, salinity, turbidity, a handful of physical chemical parameters. What we're hoping to do is take that information, use that as input to the model that Emma is working on. That says, okay, here are the conditions in these locations where you have sensors. Her model then will take those point sources of information and fill in the gaps in real time.
Geographically
Between the sensors, because we can't put a sensor every square meter, right? Even with the cheaper sensors.
Right.
You can only put them in a few spots. So you put them in a few spots, you get information in real time from specific locations, you couple it to a physical model that says, okay, we know the temperature and salinity here, here, here. The model then fills in all the gaps and allows you to get virtual complete coverage of the entire canal and the coastal zone by projecting it from a few point measurements. Once you know that entire field of temperature and salinity from the model, she plugs in the biological, and we can then predict growth rates in every position in the entire system that we're modeling.
Really smart averaging is what it amounts to between points. Knowing about the way the water is flowing, knowing about the temperature, knowing all the factors, being able to how refined is it? Is it by the inch, by the foot, by the meter?
It depends on what sort of resolution you want and how, I don't know, fancy your computers are. But in the alloy for our model, we have, I think, probably 3 points across. I'm not sure what the width of the canal is, but whatever that is divided by 3 is about what our spatial resolution is in the canal. And then as you get further offshore, it gets wider.
Sounds like every 20 feet or something along
Yeah.
those lines. So we're gonna have to close, but I'd like to get you guys to tell them when they should avoid going in the LOI. What are the signs and signals that should tell them that there might be some Vibrio out there that's waiting to make them a hamburger? Yeah.
I have a little phrase about water quality. When it rains, it's poor.
There you go. You heard it here on ThinkTech. When it rains, it's poor. That's Greg Stewart, professor of oceanography in SOEST and also in Seymour, and Emma Nuss, graduate student, working both on Vibrio vulnificus here on research in Manoa, talking about pathogens in the alloy. Most interesting discussion. Thank you so much.
Thank you for having us.
Yeah.
Aloha.
Aloha.